If I told you that the secret to healing your next paper cut is to actually force the cells in your finger to age decades in a matter of minutes, you would probably think I was completely out of my mind.
I mean, I would think you're crazy too. We are basically always taught that aging is the ultimate enemy of healing, right?
Exactly. We picture it as this incredibly slow, inevitable decay, like a vintage car left out in the elements. First, the paint fades, then a little rust appears on the bumper, and decades later, the whole chassis is just oxidized and frozen.
Yeah, and at the cellular level, that biological rusting is exactly what we call cellular senescence. For years, the prevailing dogma in science has been that these senescent, aging cells are just damaged goods.
Right, just useless junk.
Pretty much. The textbook picture is that they permanently stop dividing, they just sit around taking up space in our tissues, and they slowly leak this inflammatory junk that drives age-related diseases. Things like arthritis or Alzheimer's.
But then you step into the world of tissue regeneration and acute wound healing, and suddenly that slow-rusting car metaphor just falls completely apart. We are looking at a biological landscape today that flips everything we thought we knew about aging upside down. Welcome to today's deep dive.
It is a really exciting one today.
It really is. We are digging into a groundbreaking 2026 article from the journal Nature Cell Biology. It is titled Transcription-Independent Induction of Rapid-Onset Senescence is Integral to Healing. And our mission today is to explore how your skin uses these exact same aging cells, not as passive rusting junk, but as an elite rapid response trauma team to heal wounds almost instantly.
It is such a wild concept to wrap your head around.
It is. So, okay, let's unpack this. Because the traditional view says senescence takes days, weeks, or even months of accumulated damage to develop. But this paper completely changes the timeline. Before we get to the speed, though, how do scientists even know a cell is definitively, a senescent?
Well, in the scientific community, you cannot just look at a cell under a microscope, see that it looks a little ragged and it's declared old. You have to actually prove it biochemically.
Right.
So the researchers behind this study adhered to this very strict diagnostic checklist known as the MICS guidelines. That's M-I-C-S-E. It is basically a set of rules to ensure we are looking at a bona fide senescent cell in a living organism rather than a cell that is just temporarily stressed out or dying.
Okay, so what is the first thing on that diagnostic checklist? I imagine if a cell is aging, the first thing it does is just stop making copies of itself, right?
Yeah, that is the defining feature. The cell has to be in a state of permanent arrest. To prove this, the researchers looked for a massive spike in a specific protein called P21.
P21, got it.
You can think of the P21 protein as the emergency brake for the cell's division cycle. When P21 floods the system, the cell is locked down. But slamming on the brake isn't enough to prove senescence. You also have to show that the cell has completely taken its foot off the gas.
Oh, interesting. So what does the gas pedal actually look like in a cell?
It is a marker called Ki67.
Ki67, okay.
Right, and it indicates active proliferation. When the researchers stained these cells at the wound site, the results were incredibly stark. The cells were completely mutually exclusive. If a cell had high levels of the P21 brake, it had absolutely zero of that Ki67 gas pedal.
It was totally frozen in place.
Exactly, completely frozen.
Okay, so the cell is frozen, but that alone doesn't mean it's structurally old, right? What else is on this MICAC checklist?
Next, they look for structural collapse. Inside a normal cell, the nucleus, the vault that holds your DNA, is supported by this rigid scaffolding protein called lemon B1.
Okay.
In senescent cells, that scaffolding essentially snaps. The researchers measured a severe drop in lemon B1, meaning the nuclear envelope was actively degrading and sagging inward.
Oh, wow. So it's physically caving in.
Yeah, it's falling apart. And on top of that structural failure, they observed dramatic cell hypertrophy.
Hypertrophy, meaning they literally blow up in size.
They swell significantly, yeah. They become physically massive compared to the healthy dividing cells right next to them. And as they swell, they start exhibiting this strange behavior called the Elise phenotype.
ALAS. What does that stand for?
It stands for accumulation of lipids in senescence. Basically, they just start hoarding fat.
Wait, fat hoarding? Why would a random skin cell suddenly start packing on lipid droplets?
It's weird, right? But it is a major hallmark of the senescent state. The researchers tracked a protein called PNN2, which essentially coats the surface of these lipid droplets.
Okay.
In normal, healthy skin, you really only see PNN2 in the sebaceous glands. the parts of the skin actually designed to make oil. But at the edge of these wounds, these arrested swollen cells were absolutely packed with PN2 positive fat droplets.
So let me just paint this picture for a second. We have a cell that has slammed on the emergency brake. Its nucleus is physically collapsing, it's bloated to twice its size, and it's suddenly hoarding fat droplets.
That is the exact profile, yes.
Right. And the researchers are seeing all of this happen by making excision wounds in the skin of pigs and mice. But here is where the timeline just completely breaks my brain. If senescence is this complex structural decay, a process we normally associate with decades of wear and tear, how long did it actually take for these bloated old cells to show up after the skin was cut?
Well, in the porcine models, the pigs, these fully senescent multi-marker cells, appeared at the edge of the wound in 90 minutes.
90 minutes? That makes no sense. I mean, if senescence is like a car rusting out over 80 years, how are we seeing a fully rusted chassis appear an hour and a half after a fender bender?
It's wild, and it shatters the entire passive aging paradigm. What this rapid onset proves is that in a living physiological context, senescence is not always this slow wear and tear process caused by accumulated DNA damage.
Right.
When a tissue is violently breached, senescence is an active, programmed emergency state that a cell can just jump into almost instantly.
Okay, so let's just do a quick recap here for you listening. Our first major takeaway is that senescence involves very specific markers, right? You've got the P21 break, the low lamin B1 scaffolding, and that high PLIN2 fat hoarding. And our second point is that these cells appear at wound sites in as little as 90 minutes. So my question to you, the listener, is this. Have you ever considered that the very mechanism responsible for aging might actually be the reason you survived your last paper cut?
It really changes how you look at a simple scratch.
It really does. Think about the last time you sliced your finger chopping vegetables. By the time you found a bandage and sat back down, the cells at the very edge of that cut had already physically transformed into these bloated, fat-hoarding, ancient-looking entities. But hold on, you were losing me a bit on the biology of how they do this so fast.
Sure.
Because even an emergency response takes time to build, right? To get that P21 break protein, doesn't the cell have to go through the whole complex manufacturing process?
It usually does, which is why this discovery is so profound. To understand the cheat code these cells are using, we have to look at how a cell normally builds a protein. The standard blueprint of your DNA is locked inside the nucleus. When a cell needs a new protein, like P21, it first has to copy that specific DNA blueprint into a messenger molecule called mRNA.
Okay, and that copying process is called transcription, right?
Exactly, transcription.
Right, and once you have that mRNA copy, it travels out of the nucleus into the main body of the cell, where the cellular factories read it and build the actual physical protein. And that second step is translation. So it's transcription, then translation.
That is the central dogma of biology right there. But transcription, unwinding the DNA, reading it, copying it, is relatively slow. If you are bleeding out or suddenly exposed to pathogens, you do not have hours to wait for transcription.
Makes sense.
So the researchers found that this rapid onset senescence is entirely transcription independent.
Wait, transcription independent, they skip the first step completely. How can you build the P21 protein without copying the DNA blueprint first?
Because the blueprint is already sitting there in the cytoplasm. When the researchers analyzed normal, healthy, unwounded skin, specifically the keratinocyte cells at the surface, they found a massive high basal level of pre-existing CDKN1A mRNA.
CDKN1A, and that is the messenger molecule for the P21 protein.
Yes, exactly.
So the skin cells are just hoarding the blueprints, like fully copied, just hovering there waiting.
They are essentially stockpiling it. They do not need to waste precious time transcribing DNA after the injury occurs, the mRNA that's already manufactured. But to prevent the skin from prematurely aging under normal conditions, that pre-existing mRNA is held under strict lockdown.
How so?
It is bound by proteins that act as nuclear export inhibitors. They effectively keep it trapped or prevent it from interacting with the cellular factories.
Oh, I love this. It's like a fire extinguisher.
Oh, that's a good analogy.
Right, because here's where it gets really interesting for you listening. The cell doesn't wait for a fire to start in the kitchen to begin smelting metal, building a red canister, and mixing chemical foam. The mRNA is the fully built fire extinguisher already mounted on the wall. And the mechanical trauma of the wound, the actual cutting of the skin, is the event that pulls the pin.
That is a brilliant way to visualize it. The physical injury is the trigger that removes those nuclear export inhibitors. The pin is pulled. Instantly, all that pre-existing mRNA is unlocked and interacts with translation initiation factors. The cellular factories just kick into high gear and pump out the P21 break protein in a matter of minutes.
So my skin cells are essentially doomsday preppers.
Oh yeah, basically.
They have a bunker full of emergency mRNA ready to go, bypassing the slow DNA machinery entirely.
It is a genius evolutionary trick.
It really is. Okay, time for another quick summary. We've learned that rapid senescence doesn't rely on new transcription. Instead, that pre-existing CDKN1A mRNA is unlocked simply by removing nuclear export inhibitors. So think about this. If your skin cells are preloading mRNA for emergencies, what other invisible stockpiles might your tissues be hiding right now, just waiting for the right emergency to pull the pin?
It is definitely something that keeps biologists up at night.
I bet. But this brings up a huge question. We know how they transform instantly, but why? Once they pull the pin, freeze their own division, and swell up, what is the actual job of these rapid-onset senescent cells? Are they just like biological sandbags blocking the wound?
Far from it. To ensure these cells weren't just dead weight, the researchers used a chemical called EDU, which is a synthetic building block of DNA. If a cell is actively dividing, it eats up the EDU, and scientists can light it up with a fluorescent dye.
Okay, and what did they see?
The rapid-onset cells consumed absolutely zero EDU. They were completely frozen. But they were hypermetabolically active. Once arrested, they undergo a massive shift in what they secrete into their environment.
What are they spitting out?
It is a phenomenon known as the SASP, the senescence-associated secretory phenotype.
Okay, SASP.
Right. These frozen cells start pumping out a highly complex cocktail of pro-migratory and pro-inflammatory factors.
So they aren't sandbags. They are the paramedics calling on the radio, guiding the ambulances to the crash site.
Exactly.
Yeah.
The SASP acts as a biochemical beacon. These factors orchestrate the entire tissue repair process. They call in specialized immune cells to clear out the dead tissue and fight bacteria. And crucially, they also stimulate the neighboring healthy skin cells to migrate into the wound bed to close the physical gap. That process is called re-epithelialization.
And to prove that these senescent paramedics are actually running the show, the researchers did some incredibly precise experiments with timing, didn't they? They wanted to see what happens to a healing wound if you deliberately sabotage this rapid senescence.
They did. And their findings here clear up a lot of muddy waters in the scientific field. They use both genetic methods and pharmacological drugs to suppress these senescent cells. And they discovered that timing is absolutely everything.
How so?
If they suppress the senescence during the very early phases of healing, like right after the injury, the results were severe, wound closure was markedly delayed, the tissue simply struggled to pull itself together.
But the crazy part is what happened when they waited. If they applied that exact same suppression during the later phases of repair, let's say a few days after the injury, it had absolutely zero effect. The wound just kept healing perfectly normally.
Yes. And this is a critical insight for modern medicine. Right now, there is a massive boom in the anti-aging industry focusing on drugs called senolytics.
Right, I've heard of those.
Yeah, there are therapies designed to hunt down and destroy senescent cells in the body to theoretically keep us young and reduce chronic inflammation.
So wait, what does this all mean for practical medicine? If you were to take one of these senolytic drugs right after a major surgery, or say immediately following a car accident, could you accidentally stop your own wounds from healing?
It is a very real possibility. You would be wiping out the exact rapid response paramedic team your body just deployed to jumpstart the healing process. This timing mechanism also perfectly explains why some older scientific studies on wound healing showed conflicting results.
Oh, really? How so?
Well, scientists use different genetically engineered mouse models to study this. One popular model is called the P21DTA mouse, which works by inhibiting protein synthesis in the cell. Another is the P21 ADAC model, which forcefully induces a self-destruct sequence called caspase adipoptosis.
Okay, so basically just different ways of assassinating the senescent cells.
Precisely. Depending on which genetic assassin you use and exactly when you trigger the death of the P21 cells, you get completely different effects on the healing timeline.
Because it completely depends on whether you are taking out the paramedics while they are actively trying to save the patient, or if you take them out after they are already packing up their gear.
Exactly.
Man, that is fascinating. Okay, let's pause for our third recap. We now know that these rapid senescent cells release SASP factors to promote migration and inflammation. And we learned that early suppression delays healing, while late suppression does absolutely nothing. So, listener, Think about a time you tried to speed up a natural process. Did intervening at the wrong step actually slow you down, just like suppressing these early cells? It's all about timing.
It really is. You can't just skip to the end of the healing process.
Right, which leads perfectly into the final piece of this puzzle. Let's say the wound is closed, the skin is bridged, the emergency is over. What happens to these geriatric, bloated, fat-filled cells? Do they just hang around and cause that area of skin to age prematurely?
Well, that is the danger of the senescent state. If they stay, that pro-inflammatory SSP cocktail goes from being a helpful siren to a chronic destructive alarm. To find out what happens to them, the researchers utilized a highly sophisticated genetic tracking tool. They used P21 lineage tracing mice,
Okay.
specifically a model with a massive name. It's called the P21 Krier AR R26 TD tomato mouse.
Okay, wow, that is a massive mouthful. We are going to need to translate that out of lab speak. How does this tracker actually work?
Right, fair enough. Imagine it as a genetic tripwire connected to a can of fluorescent red paint. The system allows scientists to permanently tag a cell with a bright red fluorescent protein, that's the TD tomato part, but only if that cell turns on the P21 senescence gene during a specific window of time.
And how do they control that window?
They control it by giving the mouse a drug called tamoxifen exactly when they create the wound.
Ah, okay. The tamoxifen is like the key turning on the tripwire. So the moment the injury happens, any cell that pulls the pin on its mRNA and becomes a senescent paramedic gets permanently splashed with red dye.
Yes, exactly. It acts as an indelible biological GPS tracker. Now, in some other processes, like during early embryonic development, senescent cells do their job and then somehow miraculously revert back. They turn off the P21 brake, re-enter the cell cycle, and start dividing like normal cells again.
Wow, really?
Yeah, so the researchers needed to know if these wound-induced paramedics did the same thing. Did they revert back to normal?
And the red paint proved they don't, because the cells tagged with the red TD tomato dye never incorporated that EDU chemical we talked about earlier. They never replicated their DNA. They never divided again.
Right. They remain in a state of permanent, stable arrest.
Yeah.
But they also do not stick around to cause chronic aging. The researchers tracked the healed tissue out to 4 full weeks post-wounding, which is well after the physical hole in the skin had closed. And when they looked for the red tag cells, they found absolutely zero.
Whoa.
Zero. The P21 positive cells were entirely gone.
So how did they leave the scene? I mean, if they didn't turn back into normal cells and they aren't there anymore, the body must have forcibly removed them.
They are physically eliminated, yes. And the body does this in 2 different ways depending on where the cell is located. For the epidermal keratinocytes, the cells forming the outer surface of your skin, they are simply pushed upward and shed outward, cast off with the dead skin and the scab.
Oh, that makes sense. What about the deeper ones?
For the dermal fibroblasts, the cells deeper inside the tissue, they are removed via targeted apoptosis, which is programmed cell death. They are likely cleared away and eaten by the immune system's cleanup crews.
It is just a masterclass in biological efficiency. The body takes this incredibly potent, potentially dangerous cellular state aging and uses it as an acute life-saving tool.
What's fascinating here is how elegant this system really is.
Right. It deploys the pre-made fire extinguishers, orchestrates the entire repair operation through these inflammatory beacons, and then the very second the tissue is secured, it ruthlessly executes or ejects those exact same cells. It's a perfectly balanced mechanism to guarantee survival without risking long-term chronic inflammation.
It truly forces us to rethink what a senescent cell actually is.
It does. Okay, our final quick recap for the listener. Rapid-onset senescent cells never divide again, and they are eliminated via shedding or apoptosis after the wound closes. So, does knowing that your body actively cleans up these aging cells change how you view a healed scar?
It makes a scar look like a pretty incredible achievement.
It really does. So let's synthesize this entire journey. We've learned that skin is heavily prepared with a constant basal stockpile of mRNA. Mechanical injury pulls the pin, triggering transcription-independent rapid-onset senescence. These supposedly aging cells become the temporary directors of repair, coordinating everything through SSP. And finally, they dutifully die off or shed away, leaving restored, healthy tissue behind.
It really makes you appreciate the invisible, hyper-coordinated magic happening every single time you scrape a knee or get a paper cut.
Millions of tiny pre-loaded fire extinguishers going off, instantly turning what we thought was useless biological rust into temporary lifesavers. Next time you get a scrape, you really should just take a second to appreciate that.
It is a testament to the sheer adaptability of our cellular networks, really.
But there is a flip side to this elegance, and it is something we want you to mull over as we wrap up today. We have just learned that this beautiful rapid-onset senescence mechanism works perfectly in the skin. And the skin is a tissue that naturally and easily sheds its top layers to the outside world, physically throwing those senescent cells in the trash.
Right. But what about internal organs that cannot simply shed their surface layers to the outside world?
Exactly. Think about the heart muscle after the massive mechanical injury of a heart attack, or the brain tissue after a traumatic concussion or a stroke. If this exact same preloaded rapid-onset paramedic mechanism exists in those deep internal tissues too, where do the paramedics go when the fire is out?
If they cannot be shed, and if the local immune cleanup crews are overwhelmed by the sheer scale of the damage, those senescent cells might become permanently trapped in the tissue.
It makes you wonder, could an acute life-saving response in a young heart or brain actually become the trapped, smoldering source of chronic age-related disease decades later? The very thing that saves you at age 30 might be the exact thing that slowly ages your heart at age 70. Suddenly, that line between healing and aging doesn't look so clear-cut anymore.
The underlying mechanisms of biology rarely are simple.
We started today by talking about a slowly rusting vintage car, but it turns out sometimes the body intentionally rusts the bumper in 90 seconds just to hold the car together until you can get to safety. Thanks for joining us on this deep dive. We will catch you next time.